tuning in to Planetterrian Daily, episode 131 for July 25th, 2026. Let's see what the latest research is telling us. Genomic variants shape engineered T-cell safety, and a mouse study shows lifelong valine restriction extends lifespan. Lifelong dietary restriction of the branched-chain amino acid valine increased both median and maximum lifespan in a mouse model. The intervention preserved physical function and reduced age-related tissue changes without the severe caloric deficit required by traditional restriction protocols. Researchers tracked metabolic and inflammatory markers across multiple tissues and found sustained improvements in mitochondrial function and reduced senescence signals. The effect appears tied to altered nutrient-sensing pathways that respond specifically to valine levels. Follow-up work will test whether short-term or late-life restriction produces similar benefits. Now, shifting to a very different area of research. A science paper presents evidence that loss of epigenetic organization, rather than irreversible DNA damage, underlies much of cellular aging in the brain. Restoring epigenetic marks in mouse models reversed aspects of neuronal identity loss and improved memory performance. The work aligns with earlier reprogramming studies showing that cell identity can be reset without correcting every DNA lesion. This reframes brain aging as potentially reversible through targeted epigenome maintenance. Ongoing experiments track whether similar restoration approaches scale to other tissues. The Planetarian Spotlight notes that this science paper builds directly on earlier coverage of cellular reprogramming and partial reprogramming safety. It strengthens the case that restoring epigenetic order can recover neuronal function without fixing every DNA lesion, and supplies new molecular readouts that could serve as biomarkers for future human trials. What remains to be seen is whether these epigenetic interventions maintain long-term stability or require repeated dosing in larger mammals. You've probably heard that aging cells simply accumulate mutations until tissues fail. The emerging picture is different. Much of what looks like cell loss or infiltration may instead reflect cells losing their epigenetic instructions and drifting into new identities while staying in place. Right now, as you listen, your neurons and other long-lived cells are maintaining complex patterns of DNA methylation and histone marks that define their function. When those patterns erode, the cells can stop expressing the genes that made them recognizable without ever migrating or dying. One memorable number comes from reprogramming studies. Restoring youthful epigenetic order in mice has reversed memory deficits within weeks, even though the underlying DNA sequence remains unchanged. The practical takeaway is to watch for blood-based or imaging biomarkers that track epigenetic maintenance rather than mutation load alone. Those readouts will tell us sooner whether lifestyle or pharmacologic approaches actually preserve cell identity with age. If cell identity can be reset through epigenetic maintenance, similar principles may soon apply to engineered immune cells. A study in Science Immunology examined how inherited genomic variants influence the performance of engineered T cells used in cancer and autoimmune treatments. Researchers identified specific variants that modify both therapeutic potency and the risk of adverse effects in patient cells. The work analyzed T-cell products from multiple donors and linked variant status to differences in expansion, persistence, and cytokine release. This mechanism points to future screening steps that could match patients to safer cell products. Next steps include prospective validation in ongoing trials to test whether pre-treatment genotyping improves outcomes. A metabolite produced by gut bacteria showed a strong association with higher atrial fibrillation incidence in large human cohorts. Experimental models confirmed the metabolite directly promotes atrial electrical instability and fibrosis. The pathway offers a modifiable target through diet or microbial interventions that lower metabolite production. This finding adds to evidence that gut-derived signals can influence cardiac electrophysiology independently of traditional risk factors. Researchers plan dietary trials to test whether reducing the metabolite lowers new-onset atrial fibrillation rates. While molecular signals from the gut affect the heart, an entirely different technology is improving detection of disease in another organ. A randomized trial across multiple real-world clinics tested an AI system for classifying retinal diseases from imaging data. The AI arm improved diagnostic agreement with expert graders compared with standard clinician review alone. Performance held across varied imaging equipment and patient populations. The study measured both sensitivity for treatable conditions and reduction in missed cases. Deployment studies will now assess integration into routine workflows and effects on referral patterns. Moving from clinical tools to fundamental physics, a newly recognized magnetic state may reshape how we build devices. The 2026 Europhysics Prize recognizes the discovery of altermagnetism, a previously unrecognized fundamental magnetic state with properties distinct from ferromagnetism and antiferromagnetism. Theoretical and experimental work demonstrated that altermagnetic materials combine zero net magnetization with strong spin-polarized currents. The finding opens routes to new spintronic devices that avoid stray fields while retaining useful magnetic responses. Several candidate materials have already been synthesized and characterized. Device prototypes are the next milestone. From new materials, we shift to long-lived quantum states that could support both computing and precision measurement. Researchers measured metastable states in ytterbium ions that persist far longer than previously quantified. These states maintain coherence suitable for quantum logic operations and optical clock references. The measurements resolve a decades-old question about the practical lifetime of the states under realistic conditions. Both quantum computing architectures and next-generation atomic clocks stand to benefit from the improved stability data. Engineering teams are now incorporating the states into prototype systems. Analysis of more than 1,000 skeletons from medieval Ypres combined osteology, isotopes, and ancient DNA to reconstruct individual life histories. The data show varied migration origins, dietary differences, and social status markers linked to health outcomes. Researchers identified patterns of childhood stress and adult disease burden that differed by burial location and grave goods. The interdisciplinary project supplies a detailed snapshot of urban health in a major medieval trade center. Future comparisons with other European sites will test how trade networks shaped population health. Large population biobanks are being used to detect recent natural selection on human genomes. The approach compares allele frequencies and haplotype patterns across birth cohorts to identify loci still under selection. Signals appear at immune-related and metabolic genes, consistent with changing pathogen and dietary pressures. This method complements ancient DNA studies by capturing selection that occurred within the last few centuries. Expanded biobank analyses will refine estimates of selection strength and timing. A new imaging technique uses quantum dots to visualize light waves traveling along metal surfaces that are otherwise invisible. The method converts the evanescent fields into detectable fluorescence without disturbing the wave propagation. Researchers demonstrated the approach on several common metal surfaces used in photonics. The technique provides spatial maps at scales relevant to nanoscale optical devices. Integration with existing microscopes is expected to broaden adoption. Field trials on smallholder farms in northern Peru showed that Blanco de Piura cacao produced 1,200–1,500 kilograms of dry beans per hectare when properly managed. This output rivals widely planted commercial varieties while retaining superior flavor and aroma. The study tracked pod set, disease incidence, and bean quality across multiple seasons. Results indicate that fine flavor varieties can support both farmer income and market demand for specialty chocolate. Extension programs will now test scaled adoption. Analysis of volatile compounds from screwworm-infested wounds identified specific odor blends that strongly attract adult flies. Prototype traps baited with synthetic versions captured more flies than existing lures in field tests. The work targets the New World screwworm, a parasite that causes substantial livestock and wildlife losses. Refined baits could support area-wide suppression programs that reduce reliance on sterile insect releases alone. Larger-scale trap networks are planned for endemic regions. Laboratory and pilot tests evaluated low-cost filtration and biological approaches for stripping nitrates from private drinking water wells. Several systems achieved consistent reduction below regulatory limits without generating hazardous byproducts. Performance was measured across varying nitrate concentrations typical of agricultural areas. The methods target households that cannot connect to centralized treatment. Field demonstrations in affected communities are the next step. A gold-catalyzed chemical step enables precise, site-specific activation of anti-cancer prodrugs inside target tissues. The reaction proceeds under physiological conditions and avoids the off-target effects seen with earlier activation chemistries. Researchers demonstrated controlled release of active drug in cell and animal models. The platform expands the range of compounds that can be delivered as inactive precursors. Preclinical safety studies are the immediate priority. Before we go, keep an eye on expanded biobank analyses that will refine estimates of recent natural selection strength on immune and metabolic genes. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. One more thing, if you liked today's episode, our sister show First Principles Daily is worth a spot in your feed. Reasoning from raw materials, not analogy, on the breakthroughs that actually cut cost. It's one of the NERA Network's daily briefings, all free at neranetwork.com. The Do Positive Pod is the club for people who do something about it. Episodes and the dispatch wall at neranetwork.com/thedppod. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.